now you will hear more complete from Dr. Wehmeyer. He is a professor in the department, and he is one of the experts on this topic, and you will enjoy. He prepare you wonderful and colorful presentation. Please, Dr. Wehmeyer.
My area is dopamine, norepinephrine, serotonin neurotransmitters. I've been working for about 40 years in that area. The lectures I'm going to give you today is a beginning of a series. The lecture is on the nerve cells and the glial cells, a little more about their properties. The glial cells we don't talk about a whole lot in the course because that's not where the real action is that we're interested in, but they are very important. And I'll talk about those today a bit, and then occasionally during the subsequent lectures, we'll bring them up when important. So what I'm going to do today is talk about neurons. Dr. Byrne already talked to you about neurons in some detail, and then about glial cells, the supportive tissue of the CNS and the peripheral nervous system as well. Have questions? Just raise your hand. If you have it, probably other people do as well. So we just want to understand the properties from a functional perspective. That's what this course is all about, is the function. We are going to talk then a little bit about how we name nerve cells. There's a number of different mechanisms that evolved over the years. The nervous system has been studied beginning with Cajal and other early scientists, Spanish scientists, for a couple of hundred years. And they named these cells early on just on the basis of their shape for the most part. But now we have chemical ways that we can label the cells, which actually are more important in terms of the function. And then, as I said, I'll talk about the glial cells as well. So first, start off with nerve cell structure and properties. Dr. Byrne talked to you about 4 parts of the neuron. We can divide this up conveniently into this soma or cell body. This is what you would refer to as being what every other cell has. Muscle cells, secretory cells, endothelial and epithelial cells. They all have the same kinds of material in them as the cell body. So I'm going to talk about that first. We call it the cell body or the cell soma. Then I'm going to move on to, don't have 2 on here. 2 actually was this region right here, which I'm not going to focus on today. Dr. Byrne will talk to you about that. I want to make this simpler than going into the axon hillock. So there's the cell soma, the axon, the dendrites up here. Wasn't looking in the right place. The axon and the nerve endings, which impinge on the dendrites and the cell body itself. And when those nerve endings are to a muscle, it's called a neuromuscular junction or the motor end plate. They often talk about in physiology. So I'm going to start off with the soma. The soma is, as you know, contains the nucleus. In cells that are neurons, there's a large amount of ribosomes and rough endoplasmic reticulum that's contained in this soma. And that is referred to in the light microscopic reference as tennyl substance. I'm sure you learned about that in histology. So this region is what we call the metabolic and biosynthetic core of the neuron. Because neurons often have axons that project for 3 or 4 feet, and lots of dendrites that can go out here and cover this entire room, this is a large responsibility for this cell soma. So they're going to, all of the proteins that are going to be made And a large amount of the energy that's going to be necessary is generated in this structure, the cell soma. It contains the same kind of structures that I'm going to talk about that are in axons and dendrites later on. And those are the microtubules that are responsible for the axoplasmic, for the transport of proteins and other kinds of molecules. We have neurofilaments. Neurofilaments, you should know, are what gives neurons their structure, their support. They're like the bones of a body, the neurofilaments. They're a 3-protein, I'm not going to go into what subunits make up that protein. 3-protein molecule that is arranged in a polymerized manner to form the neurofilaments. So they're going to be very important in here, providing the structure of the cell body. And when I talk about the other parts of the neuron, I'll go into those there as well. There is also a thing called a microfilament. Microfilament is made up of polymerized actin, and microfilament is almost always related to the movement of either some kind of a subcellular structure or the actual shape of the neuron. And I'll talk about that a little bit. So there's only 3 there that we really need to worry about: the neurofilaments, the microtubules, and the microfilaments. I say, and I mentioned on the slide here, that there is a table which describes the details of their structure. That's no longer in the syllabus, so you don't have to worry about that. So on the lecture next Wednesday, I'm going to go into the function of the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi, in the process of forming these vesicles that we find in here. Because these vesicles are the same vesicles that you find down at the nerve ending that Dr. Byrne talked to you about, containing neurotransmitter. I'm going to talk about it again today. But that's where these vesicles are formed originally. We can recycle those when we get down to the nerve ending, and we'll talk about that later. But this is where they're formed. Dendrites, these processes that stick out. One of the things that we did in this particular course is we called the nerve cell that we referred to most of the time in terms of structure versus function, we call the model neuron. Because neurons, as I'm going to talk about in a few minutes, have all different kinds of structures. But in order to have someplace to start, we just label this a model neuron. And the model neuron is what's called a multipolar neuron. And has all these cell body with all of these dendrites that are radiating out. And these dendrites, as Dr. Byrne mentioned, are the increase in the area of the neuron's membrane that allows numerous synaptic contacts. And so there have been estimates of around 10,000 synaptic contacts on a model neuron like this, if it were, for example, a motor neuron, the neuron that sends processes out to contract the muscle. So there's a large amount of input here, and I'll show you that in a second. This then pretty much defines the shape of the neuron, where those dendrites are. This shows these cytoskeletal elements that I talked about, and these broken lines represent the microtubules. And why is it appropriate to use broken lines? Well, microtubules, in fact, aren't continuous. There are breaks in the microtubules. The microtubules are made up of tubulin, as you recall from your previous lectures, and they form a hollow structure that has a positive and a negative end. and the movement of substances both in the what we call the anterograde direction, out the dendrite, and the retrograde direction back down towards the cell soma is mediated by these microtubules. Again, I'll talk about this on Wednesday because I'm going to go into exoplasmic transport next Wednesday in some detail. Then these are representative of the neurofilaments. Remember, they're the ones that provide the structure for the neuron. And then we have these little wiggles in here, which are the microfilaments, which are made up of actin. So this is a representation of a dendrite. And you'll see that we have something here that's very characteristic of some neurons, and that is an outpocketing of the dendrite into what we call a spine, a dendritic spine. These have been determined to be really important in neuroplasticity, synaptic plasticity over the last 20 or 30 years, because they can change their shape. And the lectures that Dr. Byrne will give you in the subsequent days will illustrate how a change in the diameter of this spine is going to have some dramatic impact on the conduction of the signal through that particular area. So, and I've placed in this model neuron slide some of these actin filaments in there because they do change their shape. If there's anything else that we want to talk about here. We do have, I haven't drawn it in here, there are some mitochondria in dendrites, and there are ribosomes and rough endoplasmic reticulum associated with the dendrites. So what does that mean? We can make protein in these areas, in these processes. This is not true of the next thing I'm going to talk about, and that is the axon. The axon appears as if it's not capable of making any protein, the region of the axon. So we can make protein here, and there's some theories about the biosynthesis of proteins that are associated with these different regions, which may be important in the function of the cell. So this is a slide which has been stained with a silver stain or a gold stain. Heavy metal stains have an interesting characteristic of only staining specific cells. Nobody really knows why that is, but it allows us as scientists, basic scientists for the most part, and some clinical scientists, to see the structure of neurons and glia. If this stained everything, it'd all be just black. But for some reason, only this particular process took up the stain, and that's how we can see the structure of this particular dendrite. These protrusions are the spines that I talked about. And this is obviously just one dendrite. There'd be, many, many more of these that we could look at. This then shows the spine here. And this is AT, stands for axon terminal. So here is a synapse that Dr. Byrne talked to you about. And I'm going to go over a little bit more its structure in a second. And here, again, S is a spine apparatus. And then this is a nerve terminal here with the vesicles that Dr. Byrne talked to you about. Outlined that in red just to illustrate it in more detail. Okay, the axon, it's always fairly smooth. It doesn't have the rough characteristics that the dendrite has. So if you're a scientist trying to understand the function of a particular neuron, you can usually with sectioned electron microscopy find out where that axon is. Another characteristic is that it has a very acute angle here between where the axon is and the cell body, whereas the dendrites don't. They're very gradual formation, as you can see here. This region here is where the spike that Dr. Burns is going to talk to you about is initiated. And he'll go into that in some detail, this region right here. And so this is the axon. It can bifurcate and trifurcate and lead to many different, and in this particular case, motor end plates that are interacting with the skeletal muscle. It can also put off a collateral and synapse with another axon, which will be the mechanism to have feedback. And again, Dr. Burns is going to go into all the details about how that works. This is just an introduction to the structure. The axon is filled with these neurofilaments, microfilaments, and neurotubules. One of the characteristics of the axon is that the neurotubules that are in the axon have a very specific microtubule associated protein, a MAP, that I presume that you've heard about. If not, this is an introduction to that. The microtubule associated proteins, which obviously are proteins that are associated with microtubules, in the axon are made up of tau. Tau is becoming a very important protein. If you haven't seen concussion yet, you'll learn about tau there. When I talk about Alzheimer's disease at the end of the course, I'm going to talk about tau. And the characteristic that's really important is the tau that's found here, it's usually only in axons, in Alzheimer's disease, and obviously in this characteristic of professional football players where they have an increase in tau. It's found in the dendrites and the cell body as well. So this is called a tauopathy. It's a really important observation, and it relates to what causes dementia, both in the professional football players' situation and in Alzheimer's. There's a lot of evidence that amyloid is really important in Alzheimer's. The reality is tau is really important as well. And we'll get into that when I talk about that at the end of the course. So I have a cross-section here of showing you the microtubules that are these round. They're usually in clusters. We have clusters of neurofilaments, and you can see the microfilaments usually located in the axon close to the membrane, the axolemma as we call it. So that illustrates the large number of these structures that you would find in the axon. So remember, again, these black dots here are responsible for the structure of the axon. They give it its characteristic structure, its shape. Nerve endings. We have here, see, the nerve endings coming in here on this particular spine apparatus, and another one here where there's a spine. This is a modified nerve ending with neuromuscular junction. All these are points at which, and Dr. Byrne called these synapses rather than nerve endings. So there's the nerve ending, and where the nerve ending connects to the next, the cell that it's innervating is the synapse. It's become generally accepted to just call the nerve ending itself the synapse. So don't be confused by that. So as we've talked about over and over now, these are the site of connection between one cell and another. One thing that hasn't been emphasized yet, and Dr. Byrne will go into this in great detail about how this functions, is that a nerve ending can neither excite the cell that it's synapsing with, or it can inhibit it. And he's going to talk about the mechanism of that, the ionic mechanism of that particular difference in synapses. I'm going to show you a little bit about the structure of the two. So this is the characteristic of an excitatory nerve ending, where this is an excitatory synapse, as we call it. So the characteristics are that we have round vesicles here that are, a lot of these are located throughout the nerve ending. And a lot of times they're attached to microfilaments, keeps them in place. The ones that are important for the actual synaptic transmission are the ones that are down here close to this dark stuff that I have drawn in here. Because they're going to have to fuse with that membrane just as soon as the stimulus reaches the nerve ending. So they really rapidly release their neurotransmitter. And these are drawn as round vesicles containing the neurotransmitter. So that's the most important aspect of the nerve ending, or those vesicles. Remember, those came down from the soma. So we have a lot of mitochondria in the nerve endings because there's a lot of energy necessary for the pumps that are working down here. And Dr. Byrne will talk to you about those pumps. We also have these microfilaments, which are necessary for the movement, it's believed, and the tethering of the vesicles that are not right next to the release site. We have smooth endoplasmic reticulum that's within this nerve ending. Right? Have you, in other courses, learned why smooth endoplasmic reticulum is so important? Anybody tell me what that is? Calcium storage. Calcium storage. And a lot of calcium comes in from the outside in the mechanism of releasing the neurotransmitter, but a lot of calcium is released from the endoplasmic reticulum as well. And this endoplasmic reticulum is necessary to take that calcium up rapidly so that you can have a very discrete release process. So mitochondria, synaptic vesicles, smooth endoplasmic reticulum, the dense material. The dense material is the area where the release actually occurs. There are proteins there that we'll talk about on next Wednesday that are involved in that release process. On the postsynaptic side, so that's another thing that I've introduced a word that I should define. This is presynaptic, and this cell is postsynaptic. That's the way we talk about it. Dr. Byrne may have talked to you about that, but he'll really go into it in detail in his lecture today and in subsequent lectures. So in the postsynaptic cell, we can have the dense material also. Here, it's really not illustrated. It is in the next slide, as I recall. That's where the receptors that Dr. Bean talked to you about are tethered, so that they're in the location that's necessary in order to receive the neurotransmitter and propagate the stimulus. Okay, we also have here a slide showing, and this is not in the syllabus. For some reason I took it out. I can't remember why. This is exactly the same as with a excitatory synapse. The difference is the process of preparing for electron microscopy causes these vesicles to flatten. And so a scientist who is studying various areas of the brain to understand the mechanisms of whether it's inhibitory or excitatory, he can tell whether it's inhibitory if the vesicles are flattened. Otherwise, it's identical. We have dense material. We have the smooth endoplasmic reticulum, mitochondria, microfilaments, And the release of the transmitter at this site is going to inhibit the cell. This is a nice slide I like to show just to illustrate the large number of synapses on this particular motor neuron cell. And you can see these are synapses that are on the cell soma. We really can't tell where the dendrites are here. But this is illustrative, and these black dots are the synapses that are on the, and this is only one section, a light microscopic section through this. So this is obviously a three-dimensional structure, so they're going to be all over this particular neuron. This shows the difference between the excitatory and inhibitory when we do electron microscopy. But it also is really nice to illustrate the large number of vesicles that you find at a nerve terminal, the mitochondria that are prominent. Shows a little bit about the structure of these nerve terminals. Okay, that's all for the cell biology, the structure and function. But just to expand on what Dr. Byrne talked about, what I've shown here is the cell that we talked about was this multipolar cell, this model cell, with the soma and the dendrites and the axon and the terminals. That's fairly common structure characteristic of neurons, but we have others, many, many others. So for example, we have what's called a unipolar cell. So the cell body's down here, the axon's here, and the dendrites are here. Whenever we talk about any one of these, where the dendrites are is a functional thing. The nerve endings that are going to be stimulated to this cell are up here, and so this red dot illustrates where the action potential is going to be generated. Here is a bipolar cell. Again, here's where the action potential is going to be generated. This is axon, cell body, axon. So this is, for instance, found in the olfactory system and in the retina, bipolar cells in the retina. Here's one that's fairly common. That is a pseudo-unipolar cell. We call it that because its cell body is off to the side. And here we have, down here, where the stimulus is occurring. And this is common to sensory neurons. So we have pain fibers down here. We may have temperature fibers, different kinds of sensory modalities. The axon is the action potential is the spikes generated here. It goes up and leads to the central axons that are in the spinal cord. So these are different kinds of variations of shape that we talk about that's different than this one that we use to illustrate the, and Dr. Byrne is going to continue to use this particular structure as the example. So this just shows the direction that the impulse is moving in each one of these cases. How else can we name neurons other than all of this was based on shape? Unipolar, the number of processes basically, multiple processes, two processes, one process, and then this pseudo-unipolar. Well, there are other ways, and that is the early scientists that I mentioned, like Cajal and Golgi, they based it on, and they're the ones, as it turns out, that discovered that heavy metals will stain specific neurons. And so they named on shape. Others, and some of them, are named for the guy who discovered it. So we have that process. But the one that's really the most important is this one, and that is the neurotransmitter that the cell utilizes. And we'll be talking about that in the lectures later in this block. So, for example, a pyramidal cell. It's shaped like a pyramid down here. This isn't a very good pyramid, but it's the pyramidal cells that are in cerebral cortex, you have 5 layers of these cells. So that's one way. We have granule cells. They're just little itty-bitty spots. So when we talk about the cerebellum, they're going to have granule cells. There's stellate cells and so forth. So that's one of the other ways we talk about this, all based on the ability to look at the specific structure using these heavy metals. Here is based on the scientist. This is an example of a Purkinje cell. Purkinje discovered this in the cerebellum. This is the actual stained cell here. We actually have 3 of them together here. This is and this kind of shape is important because, or this kind of cell is important because this particular cell looks like it might have a huge number of dendrites radiating from the cell body. The reality is that the Purkinje cell, if you turn this direction, there'd be one process because all of these are in a single plane. So it's like I'm standing like this and I turn like this. Looks like this is the plane of the Purkinje cell. And that's because the shape of that cell dictates and is responsible for the very unique characteristics it has in the cerebellar physiology. And we'll talk about that later in the course when we talk about movement control in the cerebellum. But Golgi and Purkinje are examples of scientists who named these kinds of cells. And this is a Purkinje cell. So then the last of this particular theme is the neurotransmitter that the cell contains. So acetylcholine cells, norepinephrine secreting cells, dopamine, serotonin, GABA, neuropeptide secreting cells, glutamate secreting cells. All of these are existent in specific organization of the brain. So how do we know where they are? Well, there are two approaches. One is you can make an antibody that will specifically recognize acetylcholine. Then you use that antibody to stain slices of the brain, and you can then reconstruct the cholinergic system of the brain. The same is true for norepinephrine, dopamine, serotonin, GABA, neuropeptides. These are all important neurotransmitters that you've heard of and we're going to talk a lot about. But we now know a lot about where they are because we can use this approach of either making antibodies to these transmitters themselves. But there's another way we can go about it. It turned out to be even more effective, and that is some of these cells contain enzymes that are only in cells that make the neurotransmitter. So you can make an antibody to those enzymes and then map where those cells are. So we have a noradrenergic system, a norepinephrine system. We have a serotonin system, and so forth. And we'll talk about that in the course where that becomes important for behavior especially. So what to know about what I've just told you? Just basic structure. I don't expect you to know what the components of neurofilaments are, anything like that. Just know that neurofilaments are important, where the organelles are and generally what their function is. How we name neurons, there's these different ways. Okay, glial cells. Glial cells are the predominant cell in the CNS. There are 80 to 90% of all the cells in the CNS are glial cells. This usually surprises people. It surprised me when I first learned it. They are the basic shape of the CNS is made up of glial cells. The nerve cells are superimposed on that. So the glial cells are the supportive, physically supportive makeup of the CNS. But even as important, They're all metabolically a supportive cell because they maintain the ionic environment that all of this nerve function that you're going to learn about in the next week or so takes place. Without those glial cells to maintain that ionic milieu, none of it could happen. So they're really important. They're classified based on size and on shape. So size, we have macroglia, which there are 4 different macroglia, and we have microglia, 1 cell. What's the difference between glial cells and neurons? They are less electrically excitable. They are electrically excitable, but not their primary function. They can be depolarized. They do have a membrane potential, but that's not their function. That's why I say less electrically excitable. They don't form synapses at all. They only have one process type. They don't have dendrites and axons. They can retain their capacity to divide. And in fact, they divide upon receiving the proper stimulus pretty readily. So you can have glial cells dividing in response to some kind of an injury, for example. Nerve cells don't divide as a general rule. The reality is they do divide. But it's so rare that they divide that we talk about them not dividing. And it's only in certain regions that they appear to divide. Glial cells are identified by their size, their shape, and the type of process. But I add this because if you become a neuropathologist, or you talk to a neuropathologist, they talk about glial cells based on what the nucleus looks like, because that's what they see when they slice the brain and stain it. And the different glial cells have different nuclear morphologies. In the syllabus, I go into more detail on this. Don't worry about that detail. It's not really important at this stage. Like this chromatin density and so forth, don't worry about that. Just know that that's another way of classifying glial cells. So the macroglia are astrocytes. They look like stars. They're oligodendroglia. Oligo means few. These just have a few processes. There are ependymal cells. In the lab yesterday, I don't know where you wouldn't have seen the ependyma, but these are the cells that line the ventricles. You saw the ventricles, presumably, but you wouldn't see the ependymal cell. And Schwann cells, which are comparable to oligodendrocytes, which I'll get into in a second, are the source of axon myelin in the periphery. So astrocytes. We have 2 kinds of astrocytes, protoplasmic and fibrous. These guys are prominent in the gray matter, and these are prominent in the white matter. Fibrous are in the white matter. They have different shapes. This is a drawing that I made of a protoplasmic astrocyte that is has thicker processes. We call it murky kind of looking processes. And it is different than the fibrous, which has these thin processes. The protoplasmic astrocyte actually forms sheets around neurons. The fibrous astrocytes makes contacts with these long extensions onto blood vessels and onto some neurons and onto the glial limitans that lines the outer boundary of the CNS. And as I said, these predominate in the white matter. This just shows you this difference. See this kind of envelope type process that comes out, and this is this very, very spiky fibrous process. Physical structure, barrier between neurons. Metabolically, they maintain the ionic environment. The pH is maintained by them. And when we talk about certain neurotransmitters, they all actually remove the neurotransmitters from the extracellular space. very important function for amino acid transmitters that Dr. Waxman will talk to you about in about 2 weeks. Dr. Bean talked to you about how they can guide axons during development. And finally, this is one of their more important functions, is that they migrate and proliferate if there's injury. And they'll form what's called a glial scar some of the brains that you guys look at later on when you look at the slices, you may find glial scars. The next one we're going to talk about is the oligodendroglia. Few processes, not nearly as many as the astrocytes. These form the myelin in the CNS. And they also insulate the soma of neurons, and then they're called satellite cells. This shows you the dense staining oligodendroglia coming out here and wrapping around an axon to form the myelin sheath. They have a very characteristic that the cells that do this aren't associated directly with the axon. They're out here in the periphery, away from the axon, and they send processes out which wrap around the axon. And so you'll have a number of different oligodendroglia which will form the myelin sheath for a particular axon. So here's one, here's one, here's one, here's one. And so there'll be a number of oligodendroglia that do that. This shows the process of their doing this. I show that specifically because the Schwann cell, which forms the myelin sheath for peripheral neurons, this is all, the oligodendroglia are all CNS neurons, forms it in a different manner. It forms the same kind of a insulating membrane around the myelin, around the axon. And you can see here that we have a Schwann cell that now is directly adjacent to this axon, and it has formed the myelin sheath. And the way it does that, it squeezes its cytoplasm out as it wraps around. And that's shown this kind of a way, where it wraps around. And so each one of these particular areas of insulation is formed by a single Schwann cell. So that's the difference between the Schwann cells and the oligodendroglia. The macroglia that are lining the ventricles and the spinal cord canal are these ependymal cells. They are composed of either columnar or more flattened cells which can propel the cerebral spinal fluid down the ventricle. There is both the release of substances and the uptake of substances through these ependymal cells. In certain areas, it's more prominent than others, but it's an important mechanism. When these are situated on a basolaminar membrane, and there's blood vessels associated with that, then this is called the choroid plexus. I don't know whether you saw choroid plexus yesterday when you looked at the brains. And the choroid plexus is responsible for the formation of the cerebral spinal fluid. There's tufts of these in the ventricles. And when you look at your brains in the course, you can pick out these little tufts of choroid plexus. This just shows them in electron micrographic. And that's the choroid plexus. Last, we have the microglia. And these are really, really small cells. All the ones that I've talked about so far are derived from ectodermal region. These are derived from a mesoderm. And they're so small that you can't find them for the most part. They multiply in response to injury, similar to what the astrocytes do. And like the astrocytes, they phagocytize material. And so if you have an injury in the brain, if you slice that brain later on and try to understand what's going on, you may well find lots of microglial nuclei that are there. It's hard to find the microglia themselves. So what to know about glia? That there are more of those than neurons. They're supportive in the ways that I've talked about. They're important in a lot of different ways. Maintenance, development. If we didn't have them, obviously we wouldn't have neuronal function. They facilitate axon regeneration. We'll talk about that later in the course. Glia are the major source of cancer in the brain. It's probably one of the most important to know about. Okay, thank you very much for your attention.